Hello and welcome to another episode of the Prof series. In this episode, we'll be covering a paper by Dr. Manish Raizada. Dr. Raizada taught many different courses within the University of Guelph and studied extensively how to incorporate symbiotic life forms into common crop species. One of his projects was to incorporate an endophyte bacteria found in a volcanic swamp into different grain crop species. And we'll get started on that right now. Phosphorus is the second most limiting macronutrient for plants within agriculture. Most of the soil reserves of phosphorus exist within an inorganic form that's mined. Within plants, phosphorus is used to make DNA, RNA, and lipid membrane. Plants can obtain phosphorus from inorganic sources through acids secreted by the root system. This is how calcium phosphates are converted into usable phosphates by the plants. There are also endophytes, which are microbes that attach to the plant without causing diseases. Some species actually improve nutrient uptake. Dr. Raizada's lab previously isolated endophytes from 13 genotypes of corn. These specimens were selected from corn varieties that were grown either without fertilizer due to being wild specimens, or grown with minimal management, like several of the land races of old. In order to screen for phosphate uptake improvements, the common and annual ryegrass was used as a base host for experimentation. This is because annual ryegrass hyperaccumulates phosphorus, so the amount of phosphorus that's observed within an annual ryegrass is very visible and easy to measure. It's also closely related to corn, specifically the Zea genes. As such, these endophytes could grow on annual ryegrass to test out their effectiveness on absorbing phosphorus at a far quicker rate due to the faster life cycle of annual ryegrass. 73 endophytes were screened for this purpose, many of them from a volcanic swamp within South America. And here are the results. The annual ryegrass was grown in an environment where the only known source of phosphorus is inorganic rock phosphorus. After 4 to 5 weeks, one endophyte strain called 3F11 induced increased biomass on the root biomass of its host. This happened consistently across 4 trials. It did not, however, increase shoot biomass or the shoot to root biomass ratio on rock phosphorus, however. Hence, it seems to be mostly promoting root production within an environment with a lot of rock phosphorus. The strain 3F11 is found to be genetically identical to a population isolated from sterilized seeds of the wild species Zea nicaraguensis, a strange swamp-growing wild corn found in the base of the San Cristobal volcano in Nicaragua. 3F11 seems to have a unique capability. It not only induces more root growth, and by proxy increases the amount of acid that breaks down rock phosphorus into soluble and usable phosphorus, but the bacteria itself also decomposes the phosphorus using its own acids. This synergistic aspect seems to create the ideal environment to break down phosphorus by considerable margin. This bacteria also actively colonizes the root hairs of the annual ryegrass. In one experiment, it was found that in vitro, as well as in the field, the bacteria quickly colonize the surface of the root hairs and through the root hair cells enter in the roots themselves. Another element that seems to have been altered by the advent of the microbe is that the root hairs seem to elongate. This is likely induced by the microbe's manipulation of the plant's physiology. Based on this information, it's likely that the ability for the bacteria to combine with other organisms is conserved across all corn relatives, which include annual ryegrass. Based on this capability, it's likely able to form a symbiotic relationship with regular corn alongside many other grass species. The endophyte 3111 also seems to specifically target root hairs, and in a few cases colonize the inside of the root hairs themselves. This microbe seems to have the ability to stimulate the growth of root hairs, and it's plausible that this may even aid in preventing disease. The species seems to have a wider host range than Zea nicaraguas, able to survive not just on that species, but on other grass species as well, indicating that it can at least survive on other species of corn, and plausibly on other grain species like wheat, oats, and rye. Zea nicaraguas is considered an endangered species because artificially created forest fires or wiping out seems to be doing a big number on this corn species. This could also mean that the endophyte that the plant has a symbiotic relationship with could also be endangered. As such, both species should be conserved for future use. And that concludes this episode. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.